Yeah, good day, friends, and welcome back to the channel. We are on the third part of our discussion on the cultivation of viruses. We've talked about cultivation of viruses using animal inoculation. We've talked about the use of embryonated chick egg. And lastly, we are going to talk about cultivation of viruses using what we call organ or tissue or cell culture. Organ tissue or cell culture. Now, this is a third method that can be used for the cultivation of viruses, and this is quite important to note. Now, within this organ tissue or cell culture, there are three major methods, and the first method is called an organ culture or a cell stroke tissue culture, or what we call an explant culture. Now, for the purposes of this video, we're just going to stick to one, and that is tissue stroke cell culture. Tissue stroke cell culture. Now, this is a method in which the cells of the viruses are replicated within cells, and these cells are grown in growth media. Now, the growth media that are used to grow these cells contain a solution of, you have salt solution, sugar solution, proteins, and calf serum. This is the whole mixture that is used to grow the cells. And while these cells are growing, the viruses are inoculated into these cells and they're able to replicate within these cells. Now, even as we say tissue culture or tissue or cell culture, there are also three major methods of doing tissue or cell culture. These three major methods are: one, a primary cell culture; secondly is a diploid cell culture, which we also call a semi-continuous line; and then we have the third one, which is a heteroploid culture, which we also call a continuous cell line. Now, what are all these things? The primary cell culture, this one uses normal human or animal cells for the culture, and they grow within a limited period of time. So it's not able, we don't use them for continuous culture, meaning that these cells grow over a period of time and then they die. They are not useful for continuous culture. You can't view them over a long period of time. Examples of such cell cultures are the use of monkey kidney cells and human amniotic cell culture. These two examples are examples of primary cell cultures that can be used to produce viruses for different purposes. Now, the second type of cell culture is the use of the diploid cell culture. Now, the diploid cell culture, like I said earlier, is also called the semi-continuous cell culture. And this one is also important. It is used for the isolation of what we call fastidious viruses. Viruses that are difficult to culture are usually cultured using the diploid cell culture, which is also called the semi-continuous cell culture. It's also used to culture or to cultivate viruses for vaccine production. Now, examples of diploid cell culture are the human embryonic long strain and the rhesus embryonic cell strain. These are examples of cells which are used to do the culture in the diploid cell culture. Now, the third one under the cell culture is the heteroploid cell culture. Heteroploid cell culture, which we also call the continuous cell culture. Heteroploid cell culture, which we also call the continuous cell culture. And this is usually derived from cancer cells. Cancer cells are used and they can be cultured indefinitely. Remember that culture cells, cancer cells, they continue to grow and grow indefinitely. So if you culture viruses within these cells, they can be used continuously. And these cells can be kept, they can be stored in a deep freeze. They can be stored at minus 70 degrees in a deep freeze, and then when the scientists want to continue, they bring them out of the deep freezers and they continue to culture them. Examples of cells that we use for this kind of culture is HeLa. HeLa is very popular and it's called human carcinoma of the cervix cell. Human carcinoma of the cervix cells are called HeLa cells, and they are used for this heteroploid continuous culture. We also have Vero. Vero is vervet monkey kidney cells. Vervet monkey kidney cells, which are also used for this process. Now, what's the advantage of doing a tissue or a cell culture? The advantages is that it's relatively easier and you can use it to culture a broad spectrum of viruses, and it's quite cheap to do. Now, what are the disadvantages? The disadvantage is that the process requires a trained technician. Also, the disadvantage is the fact that because it is cancer cells that are usually being used, it cannot be used for vaccine production. Now, this brings us to the end of our discourse of cultivation of viruses. We've talked about three major ways of cultivating viruses. Talked about human inoculation in our former video. We also have a video where we talked about the use of embryonated chick egg. You can check those videos out. And this third video talks about the use of tissue or cell culture. Thank you for listening to this video. Please share our content, like our content, and have a wonderful day.